We are doing population genetics, stuff related to population migrations, gene flow between different populations, all different events that could affect the demographic history of human populations. What I do is just work with data. I'm specialized with working with the data and getting patterns and information out of it. I usually work with data that someone else generates in the lab, and then I get the data and I get to results and conclusions, or some statistical framework in order to make sense out of the huge amounts of data that are being generated. So, I don't know, 10 years ago or 20 years ago, people used to just work with one gene and then work with it, characterize it, see what it does and distribute it along a population. But nowadays, given a lot of technological improvements, you can not sequence just one gene, but sequence full genomes from a lot of individuals from the same population. And you can do that in 2 months or something. For instance, the Human Genome Project, it took 10 years to develop, and now we can sequence a human genome in a matter of months. But now the challenge is to make sense out of all that data that is being generated. And in order to do that, you need to, of course, first of all, to manage all that data. In Denmark, in the Center for Geogenetics, where we work, we have enough computer power to deal. I don't know. Right now, I'm working with tens of human genomes, and that works fine. We can do that, and we have even bigger computers. For instance, we're working with a supercomputer, and that thing is huge. I know in terms of CPUs, in terms of processors. So a regular desktop computer, a laptop would have 4 processors, and the supercomputer in DTU, it has 1,500 processors. So computer power is available. Also technological development in order to get all that data is also available. Right now the challenge is to make sense out of the data. And actually here, in the Center for Geogenetics, it's like one of the leading centers in evolutionary biology, given that we have the advantage that we have a very ancient DNA lab setup. I would take the risk and say it's the best in the world. When you do ancient DNA, you're getting the actual data of how biology looked in the past. So, given that here in geogenetics we have this one of the best ancient DNA labs, and we can get data out of it, then it's a perfect place to work with evolutionary biology. Not only humans, but I don't know, animals, plants, anything that you can get ancient DNA from. Imagine you have a population. If you just sequence those guys, you get their genomes or part of their genomes, or whatever, and from there you can get how old is that population? So when was that population established? If that population comes from an admixture event of 2 different populations, which is the ancestral population of that population you're studying? Also, how did the common ancestor of that population look like in terms of genome sequence? So imagine you can think of one whole species as a population and another species as another population. Then you can answer all sorts of questions like when was it that the 2 species diverged? When did they diverge? Then if they diverged because of a sudden geographical event, or Easter Island was colonized by Polynesians. That's already known from archaeological data. It was colonized around the 1200s, but then from the archaeological record, people has found some evidences that may point towards a contact of Easter Islanders with Native Americans. And that sounds very crazy, actually. But archaeologists have found sweet potato, they have found the bottle gourd, which are crops that are known to be native to the Americas. And archaeologists have also found Polynesian chicken in Chile in archaeological contexts that predate the presence of the Spaniards in Chile. So there's some evidence of contact between the Polynesian ancestors of Easter Islanders and Native Americans, but it's inconclusive. In principle, if you did genomics from Easter Islanders, you could get an answer. We are sequencing Easter Islanders. What we want to answer is if we can find a pattern, a genome-wide pattern that points towards a contact and a mixture, of course, with Native Americans. How would that look like? So different populations have different frequencies in different allelic frequency. An allele is just a variant of a gene. So if you have had contact with an external population, your allelic frequencies would look more like the other population. I don't know. The ideal thing would be to say, oh, it happened these many generations in the past, which corresponds to this timing, which makes sense in terms of archaeology or anthropology or whatever. So that would be like the best case. All of the DNA recombines, and the recombination mechanism depends on similarity. So we have 2 copies of each gene. Each human has 2 copies of each gene. So given that they are similar, they can pair with each other and then they can recombine. Transposons have the same thing, but the thing is that they're ends. So imagine this is, well, they have ends that could be similar to some other places in the genome, and when they find these similar sequences, they just jump because they can recombine. It's exactly the same mechanism that gives rise to this recombination. Actually, transposons are a very difficult thing to deal with in genomics, because, for instance, the way in which you sequence one genome, break it into little segments, and then you sequence those segments. But given that transposons are everywhere in the genome and they have and all of their copies are very similar, then you get a very little segment and you don't know from which transposon, from which copy does that segment comes from. So given that you don't know, you cannot use that. Many of the classical population genetics approaches make use of these repetitive elements called microsatellites. So DNA, it has four bases, A, C, G, T. So microsatellites are just repetitions of sequences. So imagine the motif is AC. So a microsatellite would be 20 times AC, and an allele of that microsatellite would be 21 ACs, or 15 or something. And yeah, as I told you, different populations would have different frequencies of these combinations. Data that someone else generates in the lab, and then I get the data and I get 2 results.